eye-centered retinotopic 2D representation into something that is more commensurate with our perceptual experience. And again, particularly for today, I will focus on how is 3D visual space represented in the brain. And that actually reminds me, I have a couple demos later, so I do have glasses. I need these back if you want to pass them around. Thanks. Okay, so what I thought I would do, since we have a bit of a mixed crowd today, I'm going to start off with a little bit of intro and background on just the basic topographic organization of visual cortex. And then the meat of my talk is going to be a series of studies we've been doing in my lab looking at neural representations of 3D visual space. So I have two fMRI studies, one looking at comparisons of how we code two-dimensional space versus position and depth, then getting a bit more detailed into whether we actually have maps in the brain of depth information. And then if there's time, I'll talk a little bit about some of the perceptual and behavioral studies we've been doing in the lab trying to link these. Then I'll also end with a summary and some open questions. Alex also informs me that philosophers don't need to have talks actually end with what we learned, that it's actually really good to talk about all the things we don't know, which in this case, we don't know a lot more than we do know. So I'll spend the first part talking about what we do know, and then just give you a giant laundry list of things that we still don't know at the end. Okay, so here's what we do know and what we've known for a while. So visual input is coded initially on the retina. Retina has a series of types of neurons, and along the back are these retinal ganglion cells. They project through the optic nerve into lateral geniculate nucleus of the thalamus, and then to the back of the brain. So first they hit primary visual cortex, which is V1, and then the information from there gradually gets projected outward into our secondary and later visual areas along both the dorsal stream and the ventral stream. And information generally gets more complex as you go to the later areas. Spatial information is preserved largely along these visual pathways into the brain. At the very basic courses sense, information in the right visual field is represented in the left hemisphere of the brain and vice versa. But this spatial information is actually much more than goes beyond these contralateral representations. And you can actually see maps of visual space that are preserved from the relationships on the image into the retina, into visual cortex. So this is one of the first demonstrations of it, or one of the most famous. This was done in macaques, in monkeys, a radioactive tracer study. So they had these tracers that were injected into the neurons of monkey primary visual cortex, and then they displayed, they flashed these checkerboard patterns, and what you can see is that the neurons, the specific patterns that are patterns of neurons activated when viewing this stimulus really preserves much of the relationships that you see in the image. Nowadays, we do this in a much less intrusive way. So standard technique with functional MRI is called retinotopic mapping. And in these studies, someone will lie in an MRI machine and stare at the center of the screen, and then you see a series of flashing checkerboards is typically the standard thing that might move around the field, or these rings that expand and contract, and then we can measure the neural activity while people are looking at these different locations of the visual field being stimulated, and you can get these nice maps of things like polar angle and eccentricity. Okay, so this is all great. And this is what lots of people do, and lots of people study spatial representations in the brain and use these retinotopic mapping techniques. But what really got me intrigued by this, and I think is particularly relevant to you guys here today, is what type of spatial location is actually being carried in those maps. The vast majority of people who study spatial representations in the brain and use these retinotopic mapping techniques, you fixate at the center of the screen, and you keep your eyes fixated at the center, and you have this flat 2D stimulus that moves around the screen. So we don't really know what type of information because we're not varying things. So a lot of what we've been doing in my lab is trying to vary things like eye position, or in this case, depth position, to try to get a better handle on what type of spatial information is being represented in these maps or other types of representations in the brain. So here are some possibilities for how we might go from that low-level basic retinotopic information, 2D information, to something more behaviorally relevant, in the case of today, going from two-dimensional representations into something that is a bit more depth-sensitive. One possibility is that spatial representations become more behaviorally relevant as you move to the later areas of the hierarchy. So maybe we start off with these flat, basic 2D representations, and maybe we get increasingly more behaviorally relevant or three-dimensional representations later on in the visual hierarchy. Sort of a similar but perhaps slightly different possibility is that maybe we have different brain regions that represent different types of information. So some brain regions might represent one type, another type, and maybe different depth cues and so forth. A non-mutually exclusive possibility is also that some types of spatial location we might find just simply are prioritized in the brain. Maybe the brain gives priority or has more dominant representations for some types of space than others. And if that's the case, then we can ask sort of a different type of question, which is, well, if the brain has preferential spatial representations, what does that mean for behavior and perception as well? Okay, so before I show the actual data, let me talk a little bit about what we can actually measure with fMRI. So one thing that we can ask is we can think of this question, what type of information, as what type of spatial location. So we can vary things like the dimensionality of space. So whether we're trying to map horizontal or vertical or polar angle or eccentricity or depth in this case. We could also vary the reference frame of visual information to look at these eye-centered or head-centered or world-centered or relative to the self versus others or objects and so forth. So we can vary in our stimuli different types of space, and then we can look at the neural response to it. And with fMRI, what we're not going to get is, on the level of a single neuron precise coding, exactly what it is. fMRI is a bit of an indirect measure of neural activity, but we can still get quite a bit of information about the representations. And so we can ask to at least a certain extent whether these representations are coarse, for example, on the level of like contralateral, one side of space versus the other, or how fine these representations are. We can also ask about type of representation. Is this information represented implicitly or explicitly? To define what I mean by an explicit representation, I simply mean the technical definition that I'm using is it linearly decodable? What that means is there information, can I look at the neuron or the voxel's response or the pattern of responses, and is there some information that's being conveyed explicitly by that pattern of neural activity that I can then read out from the pattern of neural activity to be able to decode something about the depth position, let's say. And then we can also ask beyond that, if there are these explicit representations of different types of space, are those representations organized systematically in any sort of topographic map, or are they just scattered throughout the brain or within a given region, and so forth? And we can measure, we can look for these map-like preferences in the brain using variations of that retinotopic mapping or topographic mapping technique that I showed you those pretty pictures of a moment ago. In this case, I'll walk through all the details of this later, but we use something called a population receptive field model. Okay, and I'll go through the details as we go. So tell you about a few of the studies looking at neural representations of space. Again, the first question I'll start off with is this one. So how do these, looking for these explicit representations, how do they vary for when we're talking about two-dimensional location versus position and depth? And then I'll talk a little bit more about, are these representations topographically mapped? Okay, so again, our problem is that the retinas are 2D, the world is 3D. So by definition, information about depth must be initially coded in terms of a two-dimensional difference on the retina. And then at some later point in the visual processing stream, that information gets combined into, that allows for our 3D percept. So variety of depth cues that we use to go from this 2D to 3D percept, binocular disparity, size, motion, perspective, occlusion, etc. And the puzzle, of course, is that depth is, it's extremely relevant for behavior. We need information about depth for many sorts of behaviors, for reaching, for navigation, for making distance judgments, even size and shape perception rely on a certain extent to depth. But depth information is somewhat, contradictorily, it's almost entirely ignored in studies of spatial representations in the brain. Most people who are studying spatial representations in the brain restrict to 2D. Now, of course, I don't want to make it seem like no one studies neural representations of depth at all. That's certainly not true. There are plenty of studies in animals and in humans looking at how we process those different depth cues and how they get integrated, representations of 3D shape in the brain, and even some of these more spatial representation type studies, so looking at the spatial aspects, so position and depth, and how different depth, positions, and location might be represented in the brain on both the neural level and fMRI. But all of these still fall short of what I think we would consider the testing of three-dimensional spatial representations. And so if we really want to understand, is there a representation of 3D space in the brain, we need to look at how these two-dimensional representations of space and the position and depth representations, how they compare and how they interact to ultimately form that 3D space. And for the vast majority studies, people either study 2D space or they study depth, but they don't really look at how it's integrated. So that's what we set out to do. A lot of this work, the first stuff that I will tell you about, was led by a former postdoc in my lab, Noni Finlayson. And what we did is we basically put people in the MRI machine while they are looking at different stimuli. We're using depth from disparity, so they wear the red-green glasses that you guys have, and they have them on in the scanner, and then they lie in the scanner, and through a mirror, they see the computer monitor, and we can project different images that through the glasses in the scanner, they have the percept of looking like we're stimulating different parts of three-dimensional space. So we have them, I'll show you a demo in a moment, fixate in the center of the screen, and they fixate at what's the central depth. And then we can stimulate different parts of the visual field that are either in front of that depth, equal to that depth, or behind that depth. And in the first stuff I'll show you, we just do that coarsely, just a big front and a big back. But in the mapping studies, we vary that continuously. Just as a brief side note, why are we using depth from disparity in particular? So first, it's one of the richest cues for depth, but perhaps more importantly, especially for these fMRI studies, the other cues that don't use disparity are going to be confounded with 2D stimulation. So for example, you see this ball as closer and that one is further, and that's using both size and occlusion cues. But what it means is that this ball is occupying a larger spot on the retina than that one, and that's going to be particularly important for fMRI studies, especially if we're trying to dissociate how one can decode information about depth position versus the 2D cues. So this is why, at least for the majority of the studies, we'll focus on the depth from disparity cue. Okay. So we present these in the brain. Here's a demo, so if you guys want to wear the glasses now, it doesn't matter which direction they go on. If you have it one way, something will appear front and back to other people, but you'll see both.
Is it bad to be at an angle?
It's not as good, but it should still work. I also think that this particular demo is not perfectly suited to the glasses that you're wearing, so you'll get some depth percept, but it won't be perfect. It looks better in the scanner. So again, the task is fixating on this central depth plane. People are basically looking for, when it dims. And then what you see is for certain blocks of time, different locations are stimulated for a few seconds at a time. And so these are either left or right, above or below, and in front or behind. So that one's going to be a top left, back, or front, depending on which direction your glasses are, and so forth. Hopefully you guys can see those. Okay, so that's pretty much this is a slightly sped up version, but people basically view several minutes of this, or actually almost hours of this, lying in the scanner. They get a few breaks to rest their eyes. So what we can stimulate is essentially 8 different locations, and each location can be defined in terms of its horizontal, vertical, and depth position. So for example, top left front, or bottom left right, bottom left back, and so forth. And so we record basically from the brain using fMRI, and we get the activation patterns throughout different areas of the brain while people are watching these stimuli. And then we do a particular type of analysis called multivoxel or multi-pattern, or multivoxel or multivariate pattern analysis. And what this allows us to do is try to decode the patterns of the neural activity. So a standard fMRI analysis that some of you guys may be familiar with might say something like, okay, I'm going to look at primary visual cortex, and I'll take all of the voxels, a voxel being a brain bit, all of the voxels within primary visual cortex, and I'll average across them, and maybe I'll ask, is the level of activity greater for stimuli in front versus stimuli in back? And that can give us some interesting information, but what if that particular part of the brain doesn't as a whole prefer front or behind? Maybe each individual voxel or each individual neuron has some preferences about the depth. So in that case, if individual neurons have preferences, some prefer front, some prefer behind, and so forth, what we can do is we can look at the pattern of responses across voxels, and that can allow us to gain a more fine-grained sense of the information that's being carried by that area. So that's what we do with this multivoxel pattern analysis. We look within a given region across the pattern of activation that's evoked by, let's say, those 8 different stimulus locations. And then we ask from the pattern of activation, we search through the brain and we say, okay, where can we decode information about the horizontal position? So whether it was left or right that was activated. Where can we decode information about vertical, above or below? Where can we decode information about depth, front or behind? And then we can look through the whole brain and we can compare that. So this is a map. This is some of the data we get. So these are maps of the horizontal information. I show you both cases. This first experiment, which is easiest to describe, you see a lot of information going forward more than here. That's because some regions just have this big contralateral organization. So in the second version, we looked at sort of equating for that and getting rid of the contralateral effect. What you still see is vast majority of visual cortex. So again, primary visual cortex is furthest in back there, and then you move to later visual areas as you move forward. So these are maps of where we can significantly decode what horizontal position was being stimulated. These are maps where we can decode which vertical location was being stimulated. And here are the maps for which depth location, front or behind, was being stimulated. And so what you can see is we can, in fact, decode this position and depth information across a variety of areas in the brain. But if you look closely, one thing you'll notice is if you look at these green maps, they look a little further forward than the red and the yellow maps. It looks like, the earliest visual areas are maybe not decoding depth. And so we wanted to directly compare this. So we took the average of the horizontal and vertical to get a measure of 2D information, and then we took the depth, and we basically, slightly more complicated than this, but essentially subtracted the 2 maps. And this allows us to get this combination searchlight map where the red colors are regions that have predominantly 2-dimensional representations. They really can only decode the horizontal and vertical. They don't have much information about depth. Green regions, which aren't many, maybe a little bit in these few isolated areas that may or may not be reliable, have more information about depth than 2D. And then these yellowish regions are the ones that you can think of as 3-dimensional representations. So they contain information about horizontal, vertical, and depth information. And so what seems really cool is it looks like this pattern where the information is gradually going from earliest visual parts of cortex that are primarily 2-dimensional representations into increasingly more 3-dimensional representations as you move to higher and higher visual areas, as if the representations sort of start off flat and get inflated later on. And we think this is super intriguing for a number of reasons. I'll talk about some of the implications later. Just to answer this first question, are there explicit representations of depth? Yes, there certainly are. But in particular, it seems like not in the earliest visual areas, but the depth representations get increasingly stronger as you move into the intermediate and higher level visual areas. So again, I'll talk about the overall implications and open questions later on. But the next thing that we wanted to do was say, okay, well, if we have these representations of depth in the brain, and we have these areas that seem to have representations of both 2-dimensional and depth, so maybe these putative integrated 3D representations, well, is that depth information organized systematically into spatial topographic maps like the 2D information that I talked about before? So again, we have these really well-known and well-classified 2D topographic maps throughout all of these visual areas, including these sort of later areas where we see some depth stuff in the first experiment. So are there depth maps there as well. So in order to address this, we took those standard 2D retinotopic mapping techniques and we basically modified them so that we could test whether there are position events. Very briefly, there are two main techniques that people use when they study this. Older technique is something known as phase-encoded mapping, and a former recently graduated student in my lab did a bunch of cool stuff looking at depth maps with that. What I'm going to tell you about today, though, is more recent work using a bit of a more sophisticated technique that can get us a bit more fine-grained in different types of information. And this is called population receptive field modeling. Okay, so let me walk you through what this technique does, because it's actually pretty cool. So the standard population receptive field modeling was developed to study 2D space. And so in this standard technique, you will have the visual field, the 2D visual field, stimuli sweep through it. So let's say you have like a checkerboard or a highly textured pattern, and it might move from left to right, or right to left, or top to bottom, and so forth. And so over the course of several minutes, or potentially hours, people view these different parts of the visual field as being stimulated, and then you record the activity from every neuron, or every voxel, rather, in the brain, as the subject is viewing this pattern of visual stimulation. And then you do a model-based analysis, which allows you to try to estimate for every voxel its preferred location and also its receptive field size. So what part of the visual field is it most sensitive to? So for example, let's say you had one voxel in this area. It might have a receptive field that looks something like this. So if this is the screen center, this particular voxel might be sensitive to something in the upper right visual field with roughly that extent. Another voxel might be sensitive to something in the lower right with a larger extent, and maybe another voxel is more foveal and smaller, and so forth. And so we can measure this for every voxel in the brain. And we can do the same thing for depth. So what we've been doing is modifying this technique, and rather than having the stimulation move across two-dimensional space, we use the red-green glasses, and we have stimuli move back and forth in the depth plane. And then we do that same model-based analysis, and now instead of asking what part of the 2D visual field is it sensitive to, we ask what part of depth space is it sensitive to. So maybe a certain voxel might pretty narrowly prefer front depths, another one back. Maybe some others have these more intermediate or broader representations, and maybe some voxels don't have sensitivity to depth at all. Okay, so is that technique roughly clear? Okay, so this is the stimulus that we used. I'll show you the movie in a moment. There will be a central patch. So task, again, is just fixate at that central patch, detect some dot dimming there, and then what you're going to see is a whole field of dots. They will all be at one depth plane at any moment in time, so they're moving around, but they're at a depth plane, and then that whole block of dots basically moves forward or back. I'll just let you watch it for a minute. So it should have started, depending on your glasses direction, at either frontmost or backmost, and now it's moving towards center, I believe. Yep, so now it should be at central depth. It'll start moving in another direction, probably. Give it a minute, maybe. There's some blank periods as well. I'll let you watch a minute more, and then, yep, so now it's starting at middle, and then it will either move to front or behind. You guys got the gist of it? Okay. So we do this complicated model on a very intuitive level. Again, we're basically taking for each voxel, we get its actual time course of activity, and we know what the stimulus was. And then so we make a series of models. So we might take one model that says, okay, let's pretend the voxel preferred a certain depth with a certain tuning width. What would its hypothetical time course look like? And then we compare that to the actual time course. Now, okay, let's try a different model. Let's try this part of depth space. How would it look, and so forth. And we cycle through and find the best model for each individual voxel. And then what that gives us for each voxel is we can basically say what is the preferred depth that voxel is sensitive to, and how closely tuned is that. Okay, so these are some depth maps. I'll talk about what's interesting about them in a moment, but just to let you guys see. So we can make maps then of each voxel's preferred depth. So reddish are the most front preferring, middle, and back. We can also make these maps of the tuning width, so how sort of broadly tuned versus narrowly tuned.
Can I just clarify?
Yep.
Can you just what is it that we're looking at here? I mean, what's this? What's the blob? Like the gray blob.
The brain.
That's, but it's which part?
Okay, yes, sorry. So this is this is one hemisphere of the brain. My apologies. It's pretty similar to some of the views I showed you before, but just to orient. So basically, when we're recording from the brain, we can get 3 dimensions, and then we can inflate the cortex a bit. So this is an inflated view of the brain. So these black and white lines you can think of as what would be the shriveled up parts. We basically take them and inflate them so that, so some of these, if we left them in the shriveled view, and I'll show you a couple shriveled views later, they get sort of tucked inside. So this is a standard way of sort of blowing it up a little bit, and we're not playing with actual brains, these are images, so we can do fun things like blow it up or flatten it. So this would be the left hemisphere, inflated a little bit, so occipital cortex, so the visual stuff in the back, temporal lobe and frontal stuff up there, and this would be looking at it from the outside in. We could look at the other side now, but this is basically looking from behind and outside. Yeah.
Sorry, is it surprising you're finding it now closer to the back of the head?
Yeah, so I'll talk about the results in a sec. Yeah, so this is just to orient. So there's a lot of information here, so let me just tell you what some of the most interesting things are, and then if people want to look at those images later, I can put them back up. First interesting point. These are maps. They look like sort of a whole bunch of mess. You're like, should I make anything of it? I don't know. Is it just noise? What I can say is that they're really reliable for a given individual. So just look broadly at patterns. This is one subject taking their first session, and this is a second session 6 months later. Hopefully you can sort of compare and see. Visually, they look pretty similar. Mathematically as well, so if we correlate sort of every voxel across the brain, we get really high correlations for within a subject. So at least on an individual person's level, it does seem like we're getting something real with this technique. What it means, that remains to be seen, but it seems to be reliable. Second interesting thing, and this relates to your question, if we look specifically at these tuning width maps, we see an interesting pattern. And so you might have said, oh, well, it looks like there's things happening earlier in the brain than what I showed you before. But look at the tuning width. The stuff that's happening earliest in the brain are these really wide, essentially untuned. So even though the model is technically fitting these voxels really well, what it's fitting them with is something that's a super wide tuning curve that essentially doesn't differentiate depth at all. So in the back of the brain, we get these untuned, and then it gets gradually more narrowly tuned as we move to the later areas. So it actually is quite consistent with what I showed you in the first study. It's just a different way of looking at it. Okay. Third interesting thing is if we look at the depth maps, the depth, preferred depth itself. So start off by asking, is it topographically organized into any sort of map? And when you look at this sort of hodgepodge, it's like, maybe, or is it reliable across subjects? Who knows what we're looking at? So I will tell you that for a variety of reasons, there seems to be something interesting happening, in particular in this region here, which is known as the transverse occipital sulcus. It's just a region, anatomical bit of the brain. If I zoom in on that sulcus and then look at the non-inflated view, so the actual crunchy version, what we can see in this subject looks remarkably like a map-like representation. So again, at least within this sulcus, we seem to have a gradual progression from voxels that prefer the back depths into the middle depths and into the front depths. Exactly what you might be looking for if you were looking for a map of depth. And so if we sort of take all the voxels along that line, that trajectory, and we plot those tuning curves, you can see this smooth progression spanning all of depth space. So this we thought was pretty cool. I mean, no one had ever really looked for or found maps of depth position in the brain. And this certainly seems like something interesting is going on. Now, this is one hemisphere of one subject, so who knows? But here is zooming in on that TOS region, that sulcus, across both hemispheres of all 8 of the subjects that we've looked at so far. And what you can see is that pretty much all subjects, they have something going on in this region. And most of them do show some sort of systematic progression spanning, all the different colors and moving from one to the other. But there's a lot of variability across subjects, and it's not the case that every subject's map looks exactly like that. So the precise location, the orientation of it, the extent of it, all varies considerably across subjects.
Excuse me. To go back to Susanna's question for a second about what we're looking at, I mean, it looked like that region is just dorsal, and I didn't see any counterpart that was ventral.
Yes, correct. So I am intentionally zooming in on this region here. There's stuff happening. There are a few reasons why I'm focusing in on this. Basically what I'm doing is doing the, sort of Julia Child jumping to the interesting stuff. We've done a lot of analyses across subjects, and at least for this particular design, this seems to be most reliably the place where we're getting depth information. In some subjects, we do see some map-like stuff in ventral as well, but it seems to be a little less reliable or a little less full extent of that. And I'll talk about that in a minute. But for the moment, there are theoretical reasons why we might expect why there might be something special about that, which I'll get to in a few slides. But zooming in on this is more practical. This is what our results were. So we read it, and it seemed like by and large across people, this was the place where you're most reliably seeing something map-like. And so what I've just done is, rather than show you these maps for everyone, because they get sort of complicated, and again, a lot of stuff shows up in the sulcus, what I'm doing is zooming in on this sulcus view, so basically cutting out that anatomical sulcus for each of the subjects. And so you can see that, one of the things I was going to point out is anatomically there's a ton of variability as well. So this sulcus itself tends to be sort of oriented slightly differently in different sizes in each subject. So this is a part of the brain where there's a lot of cross-subject variability, and so maybe it's not surprising that there would be variability in these maps. If people are interested later, I can show you some of our more sort of data-driven analyses for why we honed in on this area as well. But it does look like, this is the region we were most, has the most potential, at least in this particular experiment, for a map-like representation. This is all very preliminary work, I should say. I mean, we have it across 8 subjects. There's still a lot that needs to be done. So I'm going to answer this with an enthusiastic maybe. So I think that we do have some preliminary evidence for these map-like preferences. We get this depth location tuning in several visual areas. They show up across all of our different types of studies and many other people's, and I'll talk about that in a moment. Again, we're right now most excited about this potential of a depth map in the TOS region. Because most subjects are showing this tuning, there's really strong within subject reliability, even though across subjects seems to be pretty variable. The next thing I want to show you is, I sort of alluded to this before, that we are using only depth from disparity and this relative where we have them fixate at the center and it passively view these depths. So we've done a few follow-up studies. I'll just briefly summarize what we did. So some of the questions we've been asking with this are these maps sensitive to absolute versus relative disparity? So in a second experiment, so basically in the first one, they fixated at the center and depth moved relative to fixation. So it could be a map either of depth relative to where they are looking, or it could be a map of absolute depth, or relative to their eyes themselves or the head. So to dissociate that, in another experiment, we made it so that the fixation plane moved with the stimuli. So the stimuli are still moving in absolute depth, but now their fixation is moving along with it. So we get rid of these relative depth cues. And it turns out that those TOS maps disappear. In fact, when we do this technique, most of the voxels in the brain have these really raw, untuned widths. Another thing we've asked is, do these maps generalize across different depth cues, not just monocular disparity? So we did another version where we used depth from motion. Same sort of idea, but now we have these motion-driven depth cues that vary in depth. We get something with this, but they don't look like, they're not reliable. So if you take one subject's depth from motion maps and then that same subject's depth from disparity maps, they don't look the same. So in fact, it seems like most voxels, it's pretty idiosyncratic for an individual. They seem to be narrowly tuned, but either all front preferring or all middle preferring. We're still working on this, but it seems like maybe there are separate maps for the different depth cues, but we're not right now seeing a generalizable across all of depth. So seems like we have, again, these maps that are pretty reliable within a subject, maybe specific to relative depth and disparity. Now, of course, like I said, there are a lot more things we don't know, and we do know this is a really new venture that we've been doing. One of the things that I'm most excited about is getting back to the original question of 3D space. So this sort of took a step back. I said, oh, we really need to integrate 2D space and depth, and then I just showed you something that was only depth maps. But we needed to do that first to establish whether they were there. So what I'm really looking forward to in the future is trying to integrate this and maybe tweak these models so that we can model essentially a receptive field sphere in the brain and get 3D information. So stay tuned on that. Okay, so to answer the earlier question, so is there something special about TOS? You know, what about all these other regions? I mean, I showed you in the first part that we can decode depth in both dorsal and ventral stream, but we only really seem to be seeing the maps here. So is there something special that why would we expect to see it only in TOS? So let me tell you a little more about this region. So what I've outlined in purple overlaid is just an anatomical definition, literally what that sulcus is. Here now overlaid is where that sulcus lies relative to the standard visual areas, for those of you who are familiar with them. And so our depth-sensitive part of transverse occipital sulcus, this is again in just one subject, but across subjects it seems to overlap with a number of the sort of intermediate to later visual areas. Of particular note, we are certainly not the first to pose that these areas are involved in depth. Area V3A in particular comes up in lots and lots of studies across techniques, across whether it's monkeys or human studies, whether people are studying shape or depth location and so forth. We see a lot of depth information in that area. This area is also near some category selective visual areas. In particular, it's pretty close vicinity to an area that is known to be sensitive to scene stimuli called the occipital place area. And people separately, so the occipital place area responds really highly when people view scenes. In particular, scene layout and navigation. A few recent studies have looked at the role of distance in a scene, and OPA also responds there. And we have a recent study in my lab that's been looking at OPA for these depth cues as well, and it seems pretty interesting. If you notice, this dorsal region is also just very proximal to the parts of the brain that we typically think of as being important for vision for action. So, classic Milner and Goodell studies where, someone has a lesion to that part of their brain and they, can't reach and move things and put them at certain distances. So, I don't want to say that this is the only place that we see a map. Certainly, there's interesting stuff going on elsewhere, and it may be the case that at least in some subjects, or maybe other types of depth or other types of tasks, we might hopefully see other depth maps. But I don't think it's surprising that if there were one place that we'd expect to see a map, it would be there. Okay, so related question though. So I say if there were one place to see a map. Well, this is a representation of all the different places where there are known 2D maps of space. So what we're looking at is maybe there's some exciting map of depth here in the brain, but it's certainly nowhere near as extensive as these, probably like 30 maps of 2D space. And so, that also leads us to believe it does look like we might have some hint of this, that the representations are becoming more behaviorally relevant or more depth sensitive as we move later in the visual hierarchy. But it also might be the case that some are prioritized, and in particular that 2D spatial information might be prioritized. I'm going to skip through the perceptual stuff very briefly. We just asked is, spatial location, 2D spatial location is known to really be sort of prioritized over other visual features and plays a really important role in visual processing. So we asked if depth exerts the same influence, and the answer is no. In our set of behavioral studies, and I'm happy to show people later, we seem to find that perceptually 2D space is a bit more dominant over depth, and so this is the paradigm that we've been using, but we see similar things with equidistance, And some perceptual and attentional effects of depth tend to also be weaker or delayed compared to 2D space. And this kind of makes sense if you think of this hierarchy or prioritization, where maybe this fits with the idea that the earliest levels of processing are still fundamentally flat 2D and have to progress a little bit till we get to the depth stuff. But we do seem to have depth representation. Okay, so let me wrap up. So to summarize, I showed you guys a lot of data. So hopefully wasn't too fast. So here's what we know. So we know and have known for some time that 2D spatial information, 2D spatial location is represented throughout visual cortex and beyond, and there are extensive topographic maps of 2D location. What I've shown you now in the first part is that position and depth can be decoded in many of these later visual areas, and in the same areas where we have this known 2D information, so forming these possibly integrated 3D representations. We also have preliminary evidence for a topographic map of depth, but it's weaker, smaller, and less consistent than what we see for the 2D maps. And what I glossed over, but what probably many of you have known or studied before, is that in a number of situations, depth might also have weaker influences on visual perception than 2D location. Okay, so what don't we know? So we don't know a lot about the details of this. This is all very preliminary. So how fine-grained are these representations? Are all depths really represented equally? Is there some preferential treatment of some depth, something like a cortical magnification factor? What about the significance? So is that TOS map even real and reliable? If it is it linked to behavior? So if someone had damage to that TOS region, would that then have implications for their depth perception or their ability to locate things in distance? We don't necessarily know. Are there individual differences in the map, or clinical differences, or developmental differences? All these, I think, are really fascinating questions to follow up on. Beyond that, I mentioned, this work is a start towards getting at the integrated 3D space, but it's really not nailing it precisely yet. And so it is possible that what we're still looking at is within the same region, separate maps of 2D space and depth. And so what we really need to know is, are these separate maps that coexist, or are they truly integrated? I think I saw this in the conference program, and I liked this question. Is it one space or many? So there are a lot of different ways we can think of space, and we can ask about behaviorally relevant space, and we can manipulate things like reference frames and depth cues and attentional effects, and we can do that within the context of 3D space or 2D space or independent. And so what's represented in the brain, and how do all these different types of space, or maybe one single broad space, how do they interact? And I think the existence of these maps and representations might be able to clue us into another question, which is that position and depth information, is it represented more like an object feature, like color or texture, or rather than part of its location, or is it simply a less salient spatial dimension, but still intrinsically a spatial property? And maybe certain parts of the brain have representations of where depth is more feature-like, and certain parts of the brain where it's actually representing the space of it. Okay, final food for thought, especially for many of the philosophers or people that haven't thought too much about how things are in the brain. So what's the big deal about brain maps anyway? And so this is largely a rhetorical question, but just to have you guys thinking about why it might matter how something is represented in the brain. So does it matter if something's represented in these systematically organized, precise maps? Or if you have these explicit representations, but they're not organized into a map, or maybe you just have implicit representations. In this case, it seems like we do have maps of depth and possibly 3D space, but there are other examples, for example, the eye movement case, where the task relevant, what we think of as the real world, world-centered representations actually don't seem to be represented explicitly, certainly not in maps. And that's sort of an interesting brain teaser. Does it also matter if we have these maps or representations in a single region or across multiple regions, and whether those regions are sort of dedicated to that particular function, so-called modularity hypothesis, or whether they're interspersed multifunctional regions, and so forth. So this is mostly food for thought, but this is, I think, some of the things we can ask is not just, are these different types of space represented in maps, but why? might they be represented in a certain way. And of course, there might be other possibilities like maps making the brain more efficient. Maybe locating things next to each other helps for neural function. But maybe on the flip side, if we have too many different maps, that might make the brain unwieldy. And maybe maps can also tell us other things about the brain, like how we can delineate different functional areas. So, final question for all of you is I think what I'm hoping to get out of this workshop is, or what I'm hoping you guys will get out of this is what sorts of insights can we gain into our understanding of visual space from human fMRI or from learning about the brain? Not just what can these studies tell us about brain organization, but what can they then tell us about the behavior and perception or philosophical questions about visual space as well. So on that note, hopefully I didn't go too much over. Thanks to lab and funding and all of you guys.
So like geometry funding, I encourage students and please go first.
Thank you.
Yeah.
Okay. So have you think about just use 3D shape as the stimuli directly, like
Yeah.
here?
Yes. We've thought about it. We haven't done it yet. We simplified intentionally here, so our stimuli only stimulate one depth plane. So we're basically looking at the center of that. In principle, we could have stimuli themselves span multiple and just go with the center. We could also do it where rather than a blank sheet, we stimulate a particular part. And I think this is where we want to go with the next step of the 3D location is rather than stimulate the whole visual field at once, you can stimulate a particular part of the visual field and a particular depth. And in that case, maybe using these sort of spheres where they could grow in different directions might be the way to go. So no, I think it would be really interesting to do, but it was a lot simpler to start.
This is kind of related to our talk, and so in my mind, there is kind of interesting to study. Well, for example, in one study, you have the sphere, but you change its you stretch or compress it in depth,
Yep.
And in the other study, you have the same sphere without changing its shape, but changing its view distance. And I'm curious whether these two tasks will activate a different brain area, especially whether there's this associated pathway, the dorsal-ventral thing.
Yeah, I think it's a fabulous question. I don't know, but I think you or someone should run it.
I would like to.
No, I think it's a great question to ask, and I think you could do it with even more of the basic techniques. So you don't even need to get a map of it. You could pick those specific stimuli and say, okay, I'll take sort of a shallow, a small object here and the same size object here versus that there and the bigger there, and you could just sort of directly pit some of those key conditions. And I think it would be really interesting. I don't know, but based on your work, I would suspect it wouldn't be the same.
So thank you so much for that. So how does this all relate to grid cells? So as far as I understand it, grid cells are often understood as already representing in 3D and not doing doing a lot of translation work.
Yeah.
And so that suggests that we don't have to start with 2D and then build up to 3D.
Yeah. So grid cells. Grid cells in hippocampus. I assume most people are familiar with the basics of that. They're a really interesting question. So one difference that is worth noting is the grid cells, at least the classic view of grid cells, they're not so much a visual representation of space, they're more a representation of where someone is within a space. So as the rat moves to different locations and in 3D and relative to depth, you see that.
But the representation happens via visual input.
Yes. So right. So it's an interesting question how that is put together. Again, the hippocampus is typically thought of as an even later area. So we've started doing some studies in the hippocampus. We actually have looked in the hippocampus with that retinotopic versus spatiotopic. And I'm really hoping we can tap into the hippocampus and this depth stuff as well. Because I think you're right. I think we would find depth-sensitive representations in the hippocampus, but it doesn't mean it got there automatically. I think actually, contrary to that, it's even more steps to get to the hippocampus, and the hippocampus is sort of built up, at least in terms of visual processing. Right here. Come on. Okay. So, we typically think of information coming in, going to visual cortex, and then along the ventral stream. It goes along a number of visual areas, gets to even parahippocampal place area, which is near the hippocampus, doing your scene stuff, and then gets into the hippocampus. So I think it is definitely a likely place where we would expect to see maybe the full integrated 3D representations, even if they're not. Maybe we would see maps there. I think it would be super cool to look. The mapping study that I showed you, we did not have the resolution coverage to look within the hippocampus if there's a map, but I think that would be really interesting. So I would suspect it might very well be maps there. But again, I don't think that answers the question of how it got there or whether it's more intrinsic.
Okay.
I don't know if that helps.
So I'm a philosopher, and I'm begging you to look at ventral stream because we have conscious experience of depth, and it's not clear that could be explained by the TOS location.
Why not?
I hope you'll find something in ventral stream, or you'll establish a lot of connectivity.
But why would a dorsal stream area not have support a conscious representation?
The reputation of the dorsal stream is the seat of conscious, of the ventral stream is the seat of consciousness.
So not among brain imaging people.
So could you say a little bit more about that?
So if you asked me the seat of consciousness, I would definitely.
Seat of visual consciousness.
We all know it's the pineal gland. The whole question of where it is a little.
Yes, right. So I'll say a little bit about this dorsal ventral differentiation. One typical view is that the ventral stream sort of combines things into recognizable objects. So maybe if you're talking about object recognition in that sense, that might be supported more by ventral stream. I don't think that, to say that the ventral stream supports conscious processing. I mean, even, early visual cortex already is going to represent things that are sort of perception versus, what people actually see. So, or what is actually presented to the eye. So, I think that's a much more complex question. I think there is plenty of evidence, first of all, that the two streams are actually not as dissociable as the classic view. So classic view is like, okay, you only have information about object identity and object recognition along the ventral stream, and you only have this sort of, how do I act in space and location in the dorsal stream. That's not true as well. So actually, if we look at both dorsal stream areas and ventral stream areas, you can decode, you have information about object identity actually up in dorsal stream as well. You have information about object location in ventral stream. The two representations are a lot more intermixed and interconnected than I think the textbook view, likes to advocate. So just to get back to your answer, I would not, I would not assume, first of all, that some conscious perceptual processing could not be supported by something in dorsal stream. The other part of your question, though, is why don't we look in ventral stream? We have that, and we are looking. And so my answer is we have looked. If we look at this, so here we can see, for example, we can specifically localize these regions of interest in ventral stream. The LOC, for example, an object-sensitive area, and we can look at that and see what's happening. And when we do that, there is information about depth. In some subjects, we see some map-like representations in those ventral stream object areas. The reason I didn't focus on it here is just simply that they seem to be less reliable and systematic across subjects. So I'm not saying it's not there. I hope it's there. I think there's a lot of work suggesting that there's really interesting depth stuff, and maybe slightly different types of depth representations in the dorsal stream than the ventral stream, and all sorts of absolute relative or metric versus categorical. And so I think I'm definitely not trying to say that the maps are only in TOS. I'm just saying that on our first pass, that was the most reliable and the most exciting thing to look at. But certainly we're looking at those ventral areas, and I think maybe some of these other manipulations might get us better maps, or the depth from motion. We're thinking, well, maybe the motion areas, we'll see it more there. Maybe if we use Ying's objects, and actually had objects that were moving in depth, maybe that would activate the ventral stream more and we would see more depth information there. So I think it's definitely possible there's something happening there. But it could also be just that it's mostly up in dorsal, and that would still be fine for function.
There is just a request for a quick follow-up. You have 30 seconds.
Yeah, Golomb's done some single cell work where he shows that there are a lot of disparity sensitive neurons in the anterior superior temporal sulcus.
Yep.
But he can't get them on fMRI, and he thinks it's because the signal's masked by the nasal cavity.
Could be. Yeah, I mean, again, these are all imperfect studies. Yeah, like I said, I'm not saying that it's only there. So sort of the anterior temporal would be up in here, and maybe with some of the newer techniques or different types of imaging, you could have image sequences that better enable you to look specifically at those areas of interest as well. But yeah, sure. I mean, I think that there's a whole host of places there very reasonably could be depth representations, and I definitely don't want you to walk away from this thinking that I'm telling you that there are only depth representations up here and everything else is an illusion. It's just that this is the most salient place we found so far for the maps.
So I'm going to ask you a conceptual question. So when I hear that there are these 2D maps, 2D representations using 2D, are you implicitly or explicitly suggesting that these representations are, as it were, planar or spatial locations in a plane? And if you are, I want to ask you, is there another possibility for what's being represented here? So the other possibility would be that it's visual directions that are being represented, but depth indeterminate visual directions. I don't know, does that distinction make any sense to you? Is it something that you've thought about and something that
Yes to all.
Good.
So yes, so what I mean by saying that we have these 2D representations, we know that there is at least information about 2D position. The problem is that people have not tested that. So if you just put something flat on a computer monitor as a plane, you see a representation of it, you don't know if that is, if part of visual cortex that, let's say, I'm fixating here, represents to this part of the visual field. Does it represent to this part at screen depth only? Is it this part, across all of depths? Is it, maybe some have here, and then maybe the reason we have other maps are here, our depth? We don't know. So that's the problem with the past studies that have not tested that, is we know there's at least 2D information, but beyond that, it could be any of these possibilities. And that's one thing we're sort of hoping to get at more by looking at these depth studies and integrating it is it the case that, one idea is maybe you have maps that are just purely 2D depth blind, and you have depth maps that are 2D blind. Maybe you have maps that are really fine specific 3D location. I think, those are the questions that I think are most fascinating. And so we're trying to start to move in that direction. So I can't answer it now, but yes, we think about those things.
And this would have great implications functionally, depending on...
Yeah, so one of the things I really want to do the full 3D version. It's, computationally and, technique-wise, it's a monster. It's, but we think we can do it. And what I think would be really cool is to test if the early visual areas that have a really, you could make these ovals basically. So, if it has a really small 2D sensitivity, does it have a bigger depth? And then as you move later on, maybe the 2D sensitivity gets broader and the depth gets narrower, and then vice versa. I think there are all sorts of cool things, and then total implications for behavior. And so, yeah, we don't know, but I'm pretty excited to look.
Oh yeah, so I was interested in the hearing more about what you plan to look at with the non-disparity defined maps. So one issue that's gotten a lot of philosophical attention, and I'm kind of drawn to it through Robert's work, that is, what's the relation between the perception of depth in static 2D pictures and in the real world? And so you mentioned kind of motion-defined depth, but that doesn't help me so much with this particular issue. So I was wondering, have you looked at other depth cues beyond that, or
not in the fMRI study? Again, for both practical reasons and theoretical reasons, we started with the disparity. I think we can draw the most unconfounded conclusions from disparity initially. Our approach is sort of, okay, once we test the disparity, then let's look across the others. Depth from motion we chose, again, largely based on practical considerations. It was the next easiest to make it not the least confounded with 2D and still get us a vivid picture. For some of these other depth cues, we're just going to have to throw up our hands and say, you know what? We don't care if it's confounded. Let's just look at this anyway. And so, no, I think we haven't looked yet. It would be great to look.
Very fascinating talk, first of all. I listened to it with my particular expertise, which is in neural network modeling. And so if I want to build a model that can actually take some stimulus and derive some of the things that you were talking about, there are these local connectivity kernels, like a Mexican hat kernel, blah, blah, blah. The point is, my default explanation of why the brain uses maps is because the functionality of the problem requires filtering with these kernels that are concentrated around the point. And to actually implement that, you need axons, and axons take volume, and you want nearby cells to be encoding similar stimuli so that you'll have less wires.
Yeah, so that's what I mean by.
So, but then, if you have some multi-dimensional space,
yes,
and the first principal component is 2D, the second principal component is 3, the third dimension, and then you have color, motion, self-motion. There are 15 different things.
Eye position.
The optimal solution to minimize wiring length on the cortical sheet is to organize the whole thing according to the most dominant source of variation, which may well be 2D. And so many of these maps could be picking up these other features, but the cortical layout is dominated by the 2D.
Absolutely.
And so I guess, is that really a question, or does your techniques allow you to pick up on anything other than the dominant organization?
So I think that is a good question. I think the answer is yes. Now, how far beyond the dominant, I think becomes the question. So for example, I could use this technique, and within regions, I could simultaneously test, is there information about 2D position? Is there information about color? Is there information about orientation, and so forth? And I can find, we can find more than one type of information within an area. So I don't think it's the case that only the single most dominant one is what we can pick up on. But I do think, first, it is likely the case that there's some, hierarchy of organization or some, principal component type thing. And, how many components down you can actually pick up on might depend on other things like other properties of how it's represented or how fine-grained. I mean, we're also using indirect techniques, so there becomes the question of with fMRI, we can't pick up on anything that's at literally a single neuron level. So if you have a really minute representation where you have next door neurons do this, and then there's a bigger one, at some point we can only pick up, on something above a certain level, and that doesn't mean it doesn't exist. So I think with the fMRI studies and with this technique, we're getting a lot better in terms of resolution. So not only the voxels get smaller, but using these pattern analyses and model-based analyses, we can get, debatably subvoxel resolution. We can't get neuron resolution. So the best way to think of this is when we find something with fMRI, it's probably there. If we don't find it with fMRI, it does not mean it's not there. So I wouldn't want to conclude, for example, the regions that we don't seem to be showing depth. Even in the decoding, when I said, the early visual representations seem to be, if you notice I called them 2D dominant, I didn't say that they were completely absent of depth information. It is possible the depth stuff is there just at a smaller grain than fMRI. So I think we can use this to say, well, if there's one type of information that is detectable with fMRI and another that's not, it doesn't mean the one that's not isn't there at all, but I think we can draw a relative difference between perhaps the dominance of those two relative to each other. So I think it can inform to a certain extent, but can't answer it.
My comment was only about map-like.
Okay, yeah.
The map is driven by the first component. That's my hypothesis.
I think not necessarily. So the TOS, where we find the depth map, we also find 2D maps there too. So I think it depends what stimulus you're using. So if I was using a stimulus that varied in 2D, I would see a 2D map there. in addition to the 3D one. And maybe those maps are orthogonal, or maybe they're sort of different layers of it. But I think if you, with the stimuli, vary the dimension you're interested in, I think even if it's not the dominant dimension, in principle, you could still find a map of it.
I was wondering what you think about this. Do you see the fact that there is a multiplicity of maps as something that creates a puzzle, given that, like, our experience of the visual field seems to be one single visual field? Do we need an Uber map that can one of these maps to account for that, or is it just a matter of integration?
I think it's a great question. We don't know. This was so secretly one of my hypotheses, one of my dream outcomes that I thought would be really cool is, we know that we have all these different 2D maps. Well, maybe there's sort of this larger depth map that spans across them. And so it might not actually be or depth or other things. Maybe it's not 10 different 2D maps. Maybe they're just 10 different parts of, as Alex says, sort of the subsection, and there's these other overarching ones. Or, it's possible that, we don't really know. There are lots of maps of 2D space, at least. And on the surface, a lot of their properties seem quite similar. So is it the case that 2D location is so important that we better duplicate it throughout the brain because we don't want to lose, one part of the brain and lose that? So maybe redundancy helps in that sense. Or is it the case that, all these 10 different maps actually have a slightly different function? And so maybe one of them represents 2D space, in this reference frame, and another one in this one, and another one 2D space for this color or this orientation. There's a whole depth of possibilities. And so we don't know. There's not really convincing evidence that all the different maps do different things for the most part. They seem to be doing largely similar things. I mean, some are more sensitive to attention than others, or maybe some to eye movements. But I think it's a really interesting and open question, and I don't think we really know yet why there are so many maps. Or, as you said, are they actually so many, or are they all just different subsections of one big map? I don't think that's an answered question.
So before you started on the map, you
Yeah.
have this, what parts of the brain are responsive to depth. You don't want to necessarily conclude that they're about depth.
Yes. Correct.
Just because I always have to talk about color, because that's what I know. If you look at chromatically selective cells, a whole lot of them are actually involved in things like edge detection and stuff like that. They're not actually really about allowing us to see color. It's just that chromatic cues are useful for performing various other visual tasks.
Yeah.
And that's presumably a lot of what's going on there, right?
I think it could be. Yeah, it's another we don't know for sure. And I think, that very rarely do we actually see that a region only does one thing. Right. Usually it is integrated with other functions. And I think, as you put it well, sometimes the function that you think you're testing is not really what it's there for. And so this is this whole other, obviously one of the greatest limitations of at least standard neuroimaging and fMRI is we can't directly establish any sort of causal or behavioral link. So we have, just because we find, let's say, a map of depth, does that mean, first of all, that that map is actually being used functionally for behavior, or is it being used in the way that we think it's being used? And just by finding a map in the brain, I think of all this as like a first step. Like, no one has gotten to this step yet. We're at least getting to this step, but it still doesn't get us to the really satisfying answers yet.
Maybe last question.
Very short comment. I want to relate your talk to Walks in the Morning. I feel like the last point in your talk is about what can we learn from these maps. I think one major thing we can learn from all these maps is actually what's invariant in the representations. So when we talk about retinal topic map in 2D, people normally talk about the retinal topic map. If it's a topological map, we actually lose metric information a lot. Now you can ask questions whether this angle preserving, this area preserving, you can ask all kinds of differential geometry questions in this. So when you go from one map to another and so on, relate back to like Alex's talk or the talk in the first talk in the morning, is what kind of information is preserved in the visual cortex. When you go look at shapes in different distance, or you look at the table from different angles and all that stuff. I think this is what the map could really be useful for. If you extend your 3D map and look at the topological properties, that would be the
I think that's
that would relate back to a lot of the discussions.
I think that's a great point, and that's like I said, I don't think we're quite there yet, but if we can establish this, then and I think it's a really nice way of putting the sort of questions that hopefully you guys could think, okay, if there was a map, then maybe we could use that to learn to test these different theories or these different potential things that could change, and maybe that varies across regions. So yeah, I think absolutely that would be a really exciting application of this.
So we have 1 minute.
So in TOS, the map that you saw, I mean, I would guess that that the depth map has to be linked to the 2D map.
Yeah.
Right. But also, in terms of the depth map, did you see cells that responded to different amounts of depth?
Yeah. So let me read it. Okay. So we can test this, and actually I have another slide later. I'll come back. I won't click. Okay. So yeah. So we can simultaneously look at that. So and this is just one sample subject. In this subject, what this is actually plotting is the tuning sensitivity, and so you can see that some have narrower curves than others. We actually got really excited by, this was our first subject, because what it looked like across this whole area, and we can actually quantify this and look at it, is that the front depths had narrower tuning curves, and they got broader as you went back, which is very consistent with like distance perception. We got really excited about that, but it turns out that not all the subjects are showing that pattern, at least in this area. But yeah, so we can look at that information. Right now we have like a whole brain's worth of all of that information and how to look at it and decide. We're still trying to figure out the optimal ways to look at it. But yes, we do see variation. What we don't see yet is, maybe along this dimension it does this, and then along the other dimension it does tuning width or something like that. We don't see evidence for that, but it doesn't mean it's not there. We maybe just haven't looked in the right place for that. Does that answer partially?
Thank you, Julie.